Hi. So, in our last discussion, we looked at how new genes originate, and we remarked on the fact that most of the time the rate of origin of new genes is not very high. However, organisms, they have to keep up with an environment which can and often does change extremely fast, and therefore a huge question is that how exactly can organisms cope with changing environments if the underlying mechanism of change, whether in terms of birth of new genes or whether in terms of mutations, are actually much slower. So, part of this answer we have already dealt with, and part of this answer is, for example, phenotypic plasticity. Now, although we have already dealt with it, I will quickly use two of the slides that we have already looked at just to refresh your memory, and then we will get into the part that we want to discuss today. So, what exactly is plasticity? Phenotypic plasticity is the ability of one genotype to produce more than one phenotype in response to different environments, and we typically show plasticity using these kind of figures called reaction norms, where you have different environments on x axis and each line represents the trait value for a particular genotype. So, when the lines are parallel to each other, then that means that when you change the environment, the phenotypic expression is not changing. So, there is no plasticity, whereas when the lines have some kind of a slope, that means that the phenotypic expression is changing across environments, which means that there is plasticity. Now, we also saw an example, well, we saw multiple examples actually. Here is just one of them to refresh your memory. So, this is the larvae of the geometrid moth Nemoria arizonaria, and this can grow either during the spring season or during the summer season. This grows on the oak tree. When it grows during the spring season, it resembles the inflorescence of the oak flower known as catkins. So, you can see this is the larva and this is the oak inflorescence. But when the same larva, same genotype is growing during the summer, then the flowers are no longer there. At that time, the larva actually represents the twig of a oak tree. So, this is what is a special case of plasticity, where instead of having continuous variation in the trait, you have discrete variations or discrete forms. This is what is known as polyphenism. Now, one of the things that we had remarked at that point is that sometimes plasticity can vary across genotypes. So, along with these two cases of no plasticity, you can have a situation wherein for example here, the red one, when it is going from say environment A to B, the value is reducing, whereas for the other two the value is increasing. So, in other words, how an environment will how a genotype will respond to an environment will vary across genotypes. Now, if you have a situation like this, where the ability to respond to environment depends on the genotype, then that means that you should be able to select for plasticity. In other words, plasticity itself becomes a selectable trait on which evolution can now operate, or rather selection can operate, and here is a very nice example of that. So, what I am showing you over here is an organism called Daphnia melanica. So, these are very small tiny crustaceans. Typically, you find them in clear water, and Daphnia are actually a good indicator of the health of a pond or a lake, because if the lake becomes polluted, then the Daphnia typically disappears. So, it is a water quality indicator. Now, turns out that mostly these are transparent things, and because of that their internal organs, they are visible from outside, as you can see over here. This is the normal form. Now, because of this, Daphnia is a very popular model organism, because you can figure out what is happening inside the organism just by placing it under a microscope. But the same transparency also creates a problem, because the organism is now vulnerable to the effects of ultraviolet rays, particularly UVB. Now, turns out that in some populations of Drosophila melanica, When you expose them to UVB, then they produce this dark pigment called melanin. And the moment you have melanin, this is what I am showing you here on top, the Daphnia becomes opaque, it becomes dark in color, and now the UVB cannot penetrate the melanin. I mean, this is the same thing that is happening on our skin, right. Now, this phenomena happens because of the action of two genes. One is called dopa decarboxylase and the other is called ABODY. Now, the melanic forms, they obviously are better in terms of not letting UVB go through, but they have two problems. First problem is that if there are predators of crustacea, and who are the predators? Typically fishes. So, these fishes mostly are visual predators. Now, when the Daphnia is transparent, then typically it is very difficult for the predator to see them, particularly in clear water. But the moment they become melanic like this, the predator's ability to spot them increases quite a bit, and at that point they basically get eaten a lot by these predators. The second thing that happens is that production of melanin is energetically expensive, because of which the maturation rate of the melanic forms is actually slower than the non-melanic form. So, there is a cost to producing the melanin. Now, there was a very nice experiment done by two people, Scoville and Frender, in 2010, where they looked at the plasticity of melanin production in these Daphnia melanica species. So, what they did was they went to 4 lakes, high alpine lakes, in which the ultraviolet ray exposure was rather high, but it was seasonally varying. So, from these 4 lakes they picked up populations of Daphnia melanica, and 2 of these lakes were such that they the Daphnia over there had never been exposed to any predators. There were no fishes ever introduced over there. The 2 other lakes were such that Daphnia had been exposed to fishes for the last 53 in one case and 91 years in another case. So, basically long term exposure. Now, this is significant because, as I told you, these fishes are very strong predators of Daphnia. So, in many other lakes the fish had actually pushed the Daphnia to extinction, but they were able to get 2 lakes where the Daphnia and the fish were coexisting. Now, what is the beauty of this whole setup? The beauty of this whole setup is that when the all the lakes are high UV lakes, which means that in all the lakes it is better for the Daphnia to form the melanin form. But in the lakes where the melanin where the predators are present, if they form melanin, they are going to be exposed to high level of predation, whereas in the 2 other lakes ideally they should not have required the to change their melanic form, but because there is seasonal variation, therefore you expect that there will be some parts of the year when it is good to have the UV, and there will be some other parts of the year when the UV is not is a burden because it reduces the growth rate. So, accordingly, so what they did was, these 2 people, they brought these I think 13 to 17 different genotypes were collected. They brought all these to the lab and then created 2 treatments, one in which they subjected them to UV and another in which there was no UV. So, that should be UV actually, UVB, not C. Now, these are the results that they got. So, here low means no UV, high means presence of UV, and what they are plotting on y axis is the amount of melanin that is being produced. Now, first let us look at what is happening to the case when there are predators. So, you can see that there is a slight change, but this is statistically non-significant. So, for all practical purposes, when they have evolved in the presence of the predators, then they have lost their ability to modulate their level of melanin in the absence of UV. But look at what is happening to those Daphnia which have evolved without the predators. In their case, when they are getting UV, they are able to produce melanin, but when the UV is being turned off, they are able to turn down their melanin exposure. And as I told you, this plasticity is very important over here because the environment is variable, and because the environment is variable, the ability to modulate the melanin level is what is needed by this Daphnia. The environment is variable for these guys also in the presence of the predators, but over there the presence of the predators is overwhelming everything else. Okay. So, these are the two results. Number one, in the presence of predators, the Daphnia have greatly reduced their plasticity, and in this particular case these two people were also able to pinpoint what were the molecules because of which that is happening. So, they showed that this is happening because in the ones which have evolved with predators, there is constant upregulation of dopa decarboxylase and ebony, and both these proteins, when they are upregulated, they actually reduce the amount of melanin in Daphnia. So, the guys who have evolved with the predators, in them that is correlating with the fact, the upregulated level of these two molecules is correlating with the fact that they have lower amount of melanin. So, this particular example actually illustrates quite a few very interesting features about phenotypic plasticity. First, as I showed you, in variable environments where it is desirable to turn on and off or upregulate or downregulate your trait, plasticity can actually be pretty advantageous. However, the plastic expression of a phenotype many a times is costly, and when I say costly, I mean it can have negative effect on fitness, and whether a particular plasticity is advantageous or not may depend on other factors. So, just think about the fact, in the context of this Daphnia, if you evolve melanin plastically, then that will end up affecting your fitness in terms of reduced growth rate. But whether this ability to turn it down is advantageous or not depends on whether predators are there or not. If the predators are there, then this turning making the thing more melanic, that you do not really have any option in turning down the melanin, right. I am sorry, the other way around, you do not have an option in terms of turning on the melanin, because if you do, the predators are going to eat you up, right. So, that is what I mean by saying whether plasticity is advantageous or not might depend on other contexts. And when plasticity expression is costly, it is possible for organisms to lose their plasticity, which might affect their future evolution. I mean, what you saw over here, because it was costly for them to change their melanic levels, they had to maintain a constant melanic level. That is why they just ended up losing the plasticity itself. And finally, this is not something that is coming from this experiment, but plastic responses are typically fast. Sometimes they can happen within one generation or maybe 1 or 2 generations, and that is why this allows organisms to quickly respond to a phenotypic change. Now, if you have followed till this point, then you are obviously going to think that is this something that is looking like a Lamarckian inheritance, because here you have ended up inducing a variation through environment, and now you are trying to select for it. Then does it not mean that this is inheritance of acquired characters or selection on acquired characters? Not exactly, and I will tell you why not. So, this is a very famous experiment. So, this is Drosophila melanogaster, and you know that this is a diptera, which means that it has 2 wings, and the second pair of wings are modified into these balancing organs known as halteres. So, they are small tiny knob like things. However, if you take eggs of Drosophila when they are about 2.5 or 3.5 hours old, then and you expose them to the vapor of ether, the chemical, then when those eggs hatch and when the adults form, the adults tend to have, instead of 3 thoracic segments, I mean they have this extra segment over here, and more importantly, instead of the halter, they actually this halter turns into wings. Okay. So, essentially this thorax which contains thoracic segment which contains the wings, that gets duplicated, as a result of which they end up having the so called bithorax phenotype, 2 thoraxes, and more importantly they have 4 wings. Now, there was a very famous experiment done by the British embryologist Conrad Waddington, where what he did was he selected flies for producing bithorax like phenotypes. So, basically what he did was he exposed them to ether, and those flies which ended up producing the bithorax phenotypes, not all of them did, some of them did, he ended up taking those eggs and allowing them to reproduce for the next generation, and he kept on doing this repeatedly. Turned out that within 29 generations, he was able to obtain flies in the selected lines that showed the bithorax phenotype even when they were not exposed to ether. In other words, now they did not need ether to produce the 4 wings. Now, this is something that Waddington said the bithorax phenotype has now been genetically assimilated. So, what did he mean by that? So, the phenomena of genetic assimilation, as Waddington thought about it, was the process by which a phenotypically plastic trait becomes genetically determined and no longer requires the environmental stimulus to be expressed. As I said, this on the face of it looks like inheritance of acquired traits, but Waddington actually went out of his way, and he kind of looked at his entire data and did all kinds of experiments to actually show that that was not the scenario. So, I will just quote from his paper. He says that the genetic basis of the bithorax character is partly a recessive gene which causes females to produce eggs developing into bithorax phenotypes, a maternal effect, and partly a number of minor genes on both the second and the third chromosomes. Remember, this work is happening in 1942, right. So, people still do not have the ability to sequence the genes and tell you that okay, this position, that thing, and so on. But in spite of that, using the kind of techniques that Morgan and his lab had figured out, this person is able to pinpoint the fact that there are up to which chromosome these genes might exist. So, Waddington was very sure that he is not talking about a phenomena which is outside the Darwinian framework. However, there were other people who kind of interpreted it as inheritance of acquired traits, and somehow they got very aggravated by it. But then later historians and later biologists also, they explicitly pointed out that look, that is not what Waddington was trying to say. So, now this leads us to a certain concept of cryptic genetic variation, and this relates to the work of Susan Lindquist. So, what was the work? So, there is a certain mutant in Drosophila which is known as the HSP83 mutant. Now, when it is homozygous for that mutation HSP83, then that thing is not viable. So, typically you have to maintain it as heterozygotes, but the heterozygous mutants, they exhibit a large diversity of phenotypes, including deformed legs, deformed eyes, small wings, notched wings, all kinds of variations in the wings, and so on and so forth. So, Rutherford and Lindquist explicitly showed that these phenotypes could be inherited, which indicated that they had some kind of a genetic basis. Then they ended up showing that this was related with the action of the gene. The mutation was in the gene HSP90, heat shock protein 90, which they hypothesized was a chaperone protein. In other words, these are those proteins which are responsible for the proper folding of many proteins, and they hypothesized that these proteins are actually taking many genetic variants and forcing them to fold in a proper way such that the whatever variation is there is actually getting buffered. Now, why should protein folding lead to buffering of genetic variation? So, you know that the functioning of any protein depends on its structure, right, and if it is not able to form the proper structure, tertiary structure, it will not be able to function. Now, why will it not be able to form a proper structure? If let us say there are some mutations because of which the structure does not get formed. So, what the hypothesis was that the chaperone proteins, they are essentially binding with these proteins during formation and essentially kind of contorting them or kind of forcing them to form the proper structure, because of which they are able to function properly, and whatever variation is there at the ATGC level, that is not showing up at the protein structure and hence not showing up at the protein function level. Now, they said that when you mutate the HSP90, which is what is happening in these HSP83 mutants, when you mutate the HSP90, then the HSP90 cannot function as chaperones anymore. And when they cannot function as chaperones anymore, now all these little genetic variants that are there in the population, they are now going to form different things, different proteins, slightly different proteins, and all this stuff, all this variation is now going to get expressed, which in the presence of HSP90 would not have otherwise been expressed. So, this kind of genetic variation which is there in the genome, but normally is not seen at the phenotype level, this is what is known as cryptic genetic variation or CGV. Now, you can imagine that this was a mega news which said that organisms have variation which is there, but which is normally not seen. Now, people obviously were skeptic for very good reasons, but Lindquist's group, they spent a lot of time and then they were able to show the same phenomena for HSP90 in Arabidopsis. So, they did exactly the same thing, they took a inhibitor of HSP90, there they did not do, so far I remember they did not do mutation, they used geldanomycin as a inhibitor, and they showed that the moment you inhibit the effect of HSP90, all kinds of mutations start showing up. Similar stuff was observed in this particular fish. This is known as the Mexican tetra. So, this fish you can find it in two forms. There are some forms which are found at the surface of the water, close to the surface of the water, and these are the ones that I am showing here on the top, and as you can see they have nice eyes, they are regular normal fish which see things. But some of these populations of the species, they tend to migrate into underwater dark caves, and when they do that, in multiple cases they lose their eyes very fast and they become blind. Now, can you see over here that there are no eyes? So, not only are there no eyes, but the orbit of the eye and the this actually shrinks and becomes either like a point or is lost completely. So, the question is, why exactly is this happening? Now, what Rohner et al. Were able to show is that this is also related to HSP90. So, if you hinder the action of HSP90, this time they used inhibitor called radicicol. So, if you end up hindering the production of HSP90, then the variation in the eye size and the orbit size increases. So, earlier suppose it is tight like this, then the variation goes up like this. Now, if you take those individuals which had lower eye size and orbit size and you breed them with each other, then in the next generation there is a very sharp drop in the mean size of the orbit and the eye, telling you that this trait is genetically inheritable. So, now the question is that okay, this is happening in the lab when you are putting them under radicicol. Why and how does that explain why these guys are losing their eye under natural conditions? So, what Rohner et al. Showed was that if you look at the water in the underground caves, then those waters typically have much lower conductivity, and this lower conductivity, it kind of acts like a stress or something, because of which it actually ends up inhibiting or mimicking the effect of radicicol. In other words, the lower conductivity again increases the variation in the variation in the eye size and the orbit size, and there are lots of individuals produced who have very low or very small eyes, very small orbits. Now, of course, the question is that okay, this is leading to the production, but then how is the eyes getting lost? What exactly is happening because of which the eyes are getting lost? Now, that work Rohner et al. Did not cover that, but there was a subsequent paper which actually ended up showing that in the dark, obviously these fishes can no longer use their eyes for catching their prey. So, they are actually depending on certain cells and certain other sensory organs in their head for catching the fish, catching their prey. And these things, the expression of these things and the sensitivity of these things is negatively correlated with the eye size. In other words, those fishes which have smaller eye size, their sensitivity for these sensory organs is much better, and therefore, they are able to catch the fish much better, catch their prey much better. As a result, this there is a positive selection for low low eye size, and as Rohner et al. Had commented that this loss of eye for the surface fish going into going into the cave underground caves, that has actually happened multiple times. It is not a one time situation in one population, it has happened multiple times in the Mexican tetra. So, this leads us to two different but very closely connected concepts in evolutionary biology in terms of how variation is generated. One is there is one is called canalization, the other is called evolutionary capacitance. So, as we saw, there can be a lot of genetic variation or cryptic genetic variation for traits, and this variation is normally not expressed because the developmental system is very robust. We just talked about HSP90, but there can be other things also which are leading to all these variations being not expressed. And this phenomena wherein there is a reduction in phenotypic variation despite it being present either in terms of the environment or in terms of the genotype, this phenomena is what is known as canalization. Now, as you can imagine, this is a very, very important phenomena. Why? Because see, all organisms, they are being continuously exposed to all kind of environmental variation. As we saw, sometimes there can be variation small mutations etc. at the genetic level as well. Now, if the organism has no way to buffer this variation, then all everybody is going to get continuous developmental shocks. So, at some level you need some way to buffer as much environmental variations to some extent. So, that is canalization. However, there has to be a limit to the buffering. If the environment changes too much such that it becomes a pressure, it becomes a stress, then the organism needs a way to generate variation, okay. And when let us say either there are changes at the genetic level or there are changes at the environmental level, and the way by which the organisms do that, like what I showed you in the context of the Mexican tetra, that is what is known as evolutionary capacitance. So, the phenomena by which phenotypic variation can be exposed to selection in response to a cue. So, this allows for a just in time release of variation that can help an organism to face environmental fluctuations. Okay. So, you can see that phenotypic plasticity, it kind of cuts both ways, or any kind of plasticity. Sometimes you need the whole thing to be expressed and sometimes you need the whole thing to be buffered. So, you have these two phenomena of canalization and capacitance which are kind of working in conjunction with each other, and the balance between the two is what is maintaining the stability of the organism. However, there is a slight twist in the tail over here. What is it? So, turns out that there is in the body a kind of RNA known as the piRNA, and these are the PIWI interacting RNA. These are small non-coding RNAs, and they are particularly involved in the epigenetic and post-transcriptional silencing of transposons, okay, the jumping genes. Remember the ones that we talked about? We were talking about retrotransposons in our previous discussion, where mRNA to cDNA to incorporation in the genome. But along with retrotransposons, you also have lots of transposons, right, which will just take segments of the gene and randomly insert them somewhere else. So, piRNAs, they tend to control the action of these transposons. Now, turns out that HSP90 plays a very important role in the formation of piRNA. So, when you end up mutating HSP90, then the piRNA level goes down, and it has been shown that this leads to major increase in the transposon induced mutagenesis. Now, if that happens, remember what we saw in the context of Rutherford and Lindquist's work. Okay, when they mutated HSP90, they started seeing all kinds of phenotypes. They assumed that the phenotype was, the variation was already there, the genetic variation was already there, and now because of mutation of HSP90, that variation is getting expressed, which is why they call it cryptic genetic variation. But if this is correct, and people have shown that this works, then this indicates that the variation was not there to begin with. The variation got created because there was a particular form of mutation through transposon whose activity had been kept suppressed by HSP90, and now when you take out that break, lots of mutation happens, and that is what is showing up. In other words, it is not pre-existing variation, it is de novo created variation. Now, this obviously changes, you know, whether our way of thinking about cryptic genetic variation. Now, another thing that we already know, particularly in the context of bacteria, it has been shown again and again, is what is known as stress induced mutagenesis. What is that? Okay, we are not talking about, you know, mutation that is induced by, let us say, UV or some mutagen and you know, other things. That is a different ball game altogether. Those are chemical or, you know, whatever radiation induced mutagenesis. But I am talking about the fact that in many bacteria it has been shown that if you stress the microorganism out, then the mutation rate goes up. Okay. This is what is known as stress induced mutagenesis. Now, obviously, if you have that, then that is another source of variation and potential variation, and lots of people are now asking the question that how important are stress induced mutagenesis in terms of you know, as a source of variation for organisms, and more importantly, whatever we are seeing as CGV, cryptic genetic variation, can that be explained away as a stress induced mutagenesis? We do not know, people are still working on this. Okay. So, now let us just take a step back and reflect on what we have learned. So, previously we had learned about how various kinds of mutations arise. In the previous discussion, we saw how new genes arise, right. But as we remarked, both are very slow processes, and organisms very often, because of environmental changes etc., need faster solutions. What can be those solutions? Previously we have learned about epigenetic mechanisms, we had learned about plasticity, and we had learned that, you know, some of those epigenetic mechanisms, well, epigenetic mechanisms are very often the underlying mechanisms behind phenotypic plasticity, you know, that is something that we have to understand. But we had previously said that many epigenetic mechanisms are not transgenerational, although we can nowadays see more and more epigenetic changes being discovered which are transgenerational, inheritable, which means that selection can act on them. In continuation of that, we today saw that plasticity itself, okay, the ability to change itself can be subjected to evolution, and all these things are basically telling us that our, you know, genetic systems, everything that we have, all that is continuous. Oh, sorry, and we also saw that environment can also induce genetic changes. So that basically means that our genome is actually being continuously bombarded by all kinds of forces that are operating at various time scales. Okay, some are operating on the short time scale, some are operating on the long time scale, and so on. And all these changes, they are continuously leaving their marks on the genome. So you can think of our genome as a very old visitor's book on which people, the kind that they keep in various shrines or monuments, where people go and sign and say something about their feelings and so on. So this has been accumulating all these signatures over the last several years, and in the context of the genome for millions of years. And if you are able to read, if you are able to understand those patterns, then it tells you a lot of things about how that organism's genome has evolved. And when you try to put that in the context of other species, other nearby close relatives of that organism, you can see how the whole history of evolution has proceeded in the context of that lineage. You can see all those patterns written in the code of the genome. But how exactly do you see that? What are those patterns? That is what we are going to do in our next discussion. See you then. Bye.